Good morning or good afternoon, as the case may be. Hey guys, and welcome to our next part of the unit. We've talked about cells and what they're made out of. We talked about the molecules that make them up, like the phospholipids, those amphipathic molecules with a positive and negative end. We've talked about proteins that embed themselves in the membrane and allow for the fluid mosaic model of the membrane function, so they can take in food through phagocytosis, and they can have, transporter proteins do transporting things. It's been awesome. We've talked about the organelles, and today we're going to talk about how they reproduce. Always the fun conversation. We're going to talk about how they reproduce asexually, which means without sex, so maybe not the most fun conversation. And we're going to see how they do that, why they do that, leading up to the idea of sexual reproduction, which is genetics, and that is our next unit. So we will start here with how cells generally reproduce themselves, and then on Wednesday we will contrast this with sexual reproduction of cells, and that will lead us straight into genetics for Friday. So get ready for an awesome lecture on a divisive issue. We're talking about division, and that was a division joke, yeah. Anyway, let's dive in. Yeah. Recording, fun things, recording. Yes, okay, here we go. Okay, so our goals for today. Our goals for today are we are going to learn about the cell cycle, which is basically the life stages of a cell. Because, cells got to be born, they got to grow up and be, fun-loving kid cells, and then, they got to grow up and become mature, reasonable, responsible adult cells, and then they got to be family cells. So we got to talk about all those. And then we're going to talk about why they divide and, what's important about that. Maybe even how do they regulate this division process. Do we want to be dividing all the time? Nope, because I'm pretty sure that's called cancer. So we're going to learn about all that stuff and what to call all the stages and how to recognize them. So without any further ado, let us dive in. This thing is really just getting all over the place in the way, and I wish I could get rid of it, but when I do try to minimize it, it actually just it stops recording and deletes everything I've done, which is really fun to realize that you've done after you've recorded about 30 minutes. Not that that's happened. Okay, so eukaryotes versus prokaryotes. What's going on here? Okay, if you remember back, there's three main domains of life. If you remember evolution, we learned about archaeans, prokarya, and eukarya. And archaea are the ancestors, the most recent ancestors of eukaryotes, and prokaryotes are the oldest ones of them all. Pro means before, so that's kind of easy to remember. And we should know that prokaryotes do not have a nucleus or membrane-bound organelles, whereas archaeans to some extent do, and eukaryotes definitely do. And you can see up there on the upper right-hand side, we can see that comparison there. In eukaryotes, again, usually multicellular and things like that. So they're typically more complex, and the reason why is that they have differentiated various different parts of themselves to perform different functions. So unlike prokaryotes, who kind of it's one person doing all the jobs, eukaryotes are like these all the organelles working together, and they each do a specialized job, and it makes them more efficient at doing certain processes. This is not to say that eukaryotes are more evolved or better than prokaryotes. It just means that they're more recently evolved and they're more complex. But I'd like to remind you that prokaryotes, like bacteria, are dividing and reproducing way faster than we are, and they make us sick and kill us all the time, so I think they're doing just fine. So anyway, let's go. So prokaryote and eukaryote aside, let's go ahead and kind of dive in. Okay, so the big thing we're going to be focusing on during this time is the nucleus, because the nucleus contains your genetic information. That's right, your DNA. And your DNA is the basis for the regulation of protein construction, and also it contains the codes to construct those proteins in a specific way. So we're going to talk about the nucleus because it's got all that DNA in it in the form of chromosomes. So the first thing to understand is kind of going back to that surface area and volume relationship we talked about. All cells have to maintain a balance of surface area and volume. So they can't grow too big, because if they do, the surface area, which is only growing in squares, will quickly be outgrown by the volume, which is growing in cubes, right? Height, length, and width versus just height and width, length and width. And that means that a cell's mouth quote unquote, which is basically its membrane, is going to be outgrown by its stomach. And so the cell will either starve or will lyse because it'll get too big. So how do cells manage their size? Well, they could get rid of stuff, but that's not really great because if they get rid of stuff, that stuff is going to start floating around in the extracellular environment, and your body would have to break it down. And also, if they're getting rid of stuff, it's not really being constructive, it's kind of waste. So how do cells actually get rid of stuff in a useful way? So they can actually do this using the process of cell division. They actually divide to make themselves smaller and to make more cells, so kill two birds with one stone. And when they do this, it involves several different life stages. So to reduce surface area to volume ratio is one of the big reasons why they divide. Let's look closer at that division process. Okay, so another reason why cells divide is to transmit genetic information. And so if we think about why we reproduce, right, thinking back to natural selection, reproduction occurs so we can pass along successful traits that have allowed us to survive and reproduce in the environment so that our kids can keep reproducing and surviving, right? That's one of those preconditions for natural selection that we talked about with Darwin. And so this division process in eukaryotes is split into two main categories of reproduction, mitosis and meiosis. We're going to be focusing on mitosis today, which is asexual reproduction, versus meiosis, which is sexual reproduction. Okay, so you can see over there, mitosis and cell division is going to create two cells out of an original one. Those cells are identical to each other. And that's the whole goal, is to just make more cells with the extra stuff you got. And both cells that are created are going to have a smaller surface area to volume ratio than the original cell. In meiosis, we create more cells, and these cells are not genetically identical. So you can kind of tell that by the pictures of the chromosomes down there. They're all kind of distinct. And meiosis is where we start getting all of the differences in traits between parents and offspring, why offspring are not exact clones of their parents. We'll talk about that in a little bit. But this highlights the second reason why we would divide, is not just to keep up our surface area and volume ratio, but also to transmit genetic information. So let's talk about the process. So the process of a cell is basically its life cycle. You can think of it like the cell's birth, childhood, adolescence, and adulthood. And I like to think of it that way because it helps it to be more relatable to me. Everything before adulthood, which basically for a cell is when it becomes a parent or splits into two new cells or more, everything before that is known as interphase. So we think of that because cells in interphase, they're kind of in between divisions. And so they're kind of in between generations. Just like you and I, we're in between generations. I mean, I'm not having kids yet. You're not having kids yet, I assume. No judgment if you are. We're not having kids currently, and so we're kind of in between generations. We are the children of the previous generation, but we haven't yet created new ones, so we're in between. And that's what interphase is, and it consists of three different stages, which I like to think of as birth, childhood, and adolescence, but really is called G1, S, and G2. G1 and G2, the G stands for gap. And essentially, when you think about a gap, you should think about sort of a space where nothing is really happening. And the gap is, something's happening, but the gap is where not a lot of activity is occurring. The gaps are just times when the cell is growing, right? So I also like to think of it as growth 1 and growth 2. So basically, the cell is just getting bigger. It's making more membrane. It's making more organelles and cytoplasm. It's just getting bigger, just like you do when you grow up. When you're a kid, you grow up. So in G1, we essentially just have us getting bigger and duplicating stuff that we will need soon. Nothing genetic is duplicated, though. It's just the organelles and the cytoplasm. And S is where we actually get the duplication of our chromosomes, so our DNA. So we make a copy of our entire genome, our entire DNA. And the reason for this is that, each new cell will have to have a whole copy. So we have to actually make 2 copies so that each of the new cells can get 1 each. That's what's happening during S. I like to think of that as your childhood. You're kind of growing up. You're making new information. You're making new knowledge, new DNA. I don't know. Yeah, just stick with my analogy, okay? And then G2 is when we get to adolescence, right? And so this is when the cell starts to strain its surface area to volume interactions, and it gets all angsty and starts listening to Green Day, and it's like anyway. So you get the point. So G2, it's growing bigger. It's got all the stuff it needs now to divide. It has like 2 of every organelle. It's ready to go. It has 2 of all of its DNA because of the S cycle. And we're ready to start division, right? So you're ready to start a family. Hopefully, well, I'm not ready. Still not ready yet for that. Some people are. Anyway, so then it leads us to adulthood. And adulthood is marked by reproduction, okay, and cells. And I guess somewhat in people. I don't know. But anyway, mitosis is the one we're going to look at here, but it could also be meiosis depending on what's happening. And mitosis is where we actually go through the process of splitting up all of that extra stuff we've been growing throughout interphase. So we split it into 2 equal pieces, so 2 new cells with an equal amount of stuff. And then after we've split the genetic information in mitosis, we go ahead and we split the organelles and cytokinesis. So mitosis is just that genetic information being split, and cytokinesis is the organelles in the cytoplasm. So all of this is extremely highly regulated. Why? Okay, so just think of it as in people, there's a reason why we say that legal reproductive age is at 18, because back in the Middle Ages, it was like 12, and a lot of people died. So in childbirth, because their bodies were physically just, not able to handle the birth of a child that was relatively large compared to their body size. So just in the same way that we make it so that, hey, you got to be 18 when you're a human, in cells, it's like, hey, you gotta have enough stuff. You gotta have enough organelles. You gotta have enough cytoplasm. You gotta have enough DNA. If you don't have that stuff, you're not ready to have kids. Just like we say, you gotta have support and know-how and be able to support yourself. All that, you get it. You get the analogy. Anyway. Okay, so we can see G0 is an additional phase. It's not considered part of interphase, but it is a sort of stasis that the cell can go into if something wrong happens at G1. And actually, to tell you the truth, there's multiple checks in G1, S, and G2 that this just isn't showing. And you can kind of see a couple of arrows branching off that show the cell stopping at a certain point, and that represents a checkpoint that the cell has to pass in order to continue to divide. Good golly. Okay. Sorry about that. Thank you for those of you who just said bless you. Aha. Okay. So, about 90% of a cell is spent in interphase. Just like we spend most of our lives living and not reproducing, most of us, I assume, cells don't spend most of their time reproducing. If you think about it, it makes sense, right? They have to have enough stuff to be able to reproduce. I mean, for us, it's a little easier. We can just, we can go out and do reproductive things all the time, all day long. It doesn't require us to actually divide in half, but it does for the cells. So, the cells, they really have to have enough stuff to divide. So I think we've talked about this enough. Here's a little summary in words if you want to pause the video and take a look at it. Otherwise, here we go. All right, and yeah, here we go. So the G0 phase, it will no longer continue to divide, and this is because it hasn't passed the checkpoint. So it's been scanned by the body and been determined to not have enough genetic information or whatever. It's not ready to move on to the next phase, and so the body kind of holds it back. Just like, I guess, if somebody wasn't ready to pass high school, the tests would kind of hold them back and say, hey, you need to spend some more time on this stuff before you move on. Right, so, and there are lots of cues that these follow, and again, G0 is not the only time this happens. It happens a lot. There are checks at every single stage. Okay. Oh my God. Oh man. Okay. Mitosis. So it's the nuclear division of the cells with the purpose of allowing the cell to be able to reduce its surface area to volume ratio, to repair tissues. So when you get a cut, you have a bunch of cells that are around that cut. You need to fill in the gap you just made. They will divide by mitosis in order to fill that gap. And then, of course, asexual reproduction. So to make sure you have enough cells that are alive and healthy in your body. In fact, your cells, it depends on what organs they're a part of and what part of your body they're a part of, but actually your cells are constantly replenishing themselves. And you actually are made out of, like, maybe at this point, probably less than 1% of your cells that you have in your body right now are the same cells that you had when you were 8. So you're actually an entirely new person. But that doesn't seem that way because your cells have been dividing via mitosis, and each of the new cells is identical to the last one, and so they're upholding the same function. So to you, it all seems like, hey, I'm the same person, but you're actually not. You are physically, literally, a different person than you were before, and you will be a wholly different person again in another decade or so. And that's pretty incredible to think about. Anyway, so hopefully I haven't just caused a major existential crisis, but that's an overview of mitosis there. Moving on. Okay, so this happens in 4 phases. And the 4 phases of mitosis are things that you're just going to have to memorize. So let's take a look. So when mitosis is ready to happen, it's going to go through something called PMAT, and it has to go through these in this particular order. This is the order by which the genetic information is split. We start with prophase, where we get a bunch of the chromosomes actually start to condense. Now, what is a chromosome? Okay, so your DNA is not always in the double helix form inside your nucleus. In fact, it's very rarely in that form because that's the form that's used when the DNA is actually being used. But funnily enough, not all of your humongous amount of DNA is being used all at once. And so most of it is coiled up and stored almost like you would store yarn, like in a little ball or something. But we call these coils, these tight coils of DNA, chromosomes. And your body has a specific number of them based off your species. So, for humans, we have 46 chromosomes. But an onion might have, thousands of chromosomes. So every species has its own number of chromosomes. And that just refers to the number of those double helix strands that we actually have. So DNA isn't all like one strand. It's not all looped together like one strand of spaghetti in a bowl. It's multiple strands, all mixed together. But then once they separate into chromosomes, once they get all wound up, you can tell that they're distinct. So prophase is when we pack up all of our chromosomes. So no more DNA stuff is really happening. We've wound up all our DNA and prepared it for separation. Metaphase is when we're going to go ahead and line up all of those DNA chromosomes to make sure that we have them all accounted for, and so we can start to divide. Now, what are those lines that are coming down to actually start to divide them? Those lines are protein filaments, and they're being created by those little yellow organelles on either side of the cell called centrioles. So these centrioles right here are going to spin out those little protein fibers like Spider-Man shoots out a web, and it's going to catch the chromosomes at the center here in this little place called the centromere. The centromere is just what holds the 2 copies of every chromosome together. Remember, during the S phase, you created a copy of each chromosome. So here they are. Here's the chromosomes. This is one chromosome. And here's its copy attached to it via a centromere. So it's basically going to grab both sides or both copies by the centromere. Oh, and if you're wondering, this over here in the prophase, this is the nucleus going away, because obviously we can't have the nucleus in the way if we want to shift these chromosomes around. So we're going to attach our spindles onto the centromeres, and then we're going to start pulling them apart in anaphase. So I like to think about anaphase, A for away, anaphase. We start pulling those chromosomes apart. We can see one of each goes into each cell, so each cell will have an identical amount of chromosomal information, one of each copy. And then we have telophase, which is when we start to actually split our cell a little bit. Notice that the cytoplasm isn't completely split yet because that happens during cytokinesis. And we see the centrioles start to dissolve and go away. And our DNA starts to turn back into its more useful ladder helix form. And the nucleus starts to come back too. So starting at the beginning, prophase, pro means before, right? So before division, we get rid of the nucleus and we start to spin our little fibers out here. Metaphase, M for metaphase, we line it up in the middle and we prepare it to divide. Anaphase, A for away, we split up all the chromosomes so we have an equal amount in each new cell. And telophase, we have 2 new nuclei, so T for 2, and those nuclei start to reform and the cell starts to divide its cytoplasm. So those are the 4 steps of mitosis, and at each step there are certain checkpoints that have to be met, otherwise the whole process of division halts and the cell might be destroyed or something might need to be fixed before it can continue. Here's a picture of some actual micrographs of these that have been done with dyed fluorescent proteins. We can see here these are those filaments that are going to start pulling it apart. This is the cell and interface, so it's not actively dividing. Prophase, we can start to see those chromosomes begin to condense, those little wormy looking things in there. Here, pro-metaphase, that means they're lining up for metaphase, but they're between prophase and metaphase. And then, of course, we start going through the rest of the division process there. So it's pretty cool to take a look at what it actually looks like. And down here we can see, this is a 3D model of the same thing happening. Yeah, so pretty cool. And here we have metaphase. They're lined up in the middle. Anaphase, they're being pulled apart. And then telophase, they're coming back together again. So we call this the equatorial plate or the metaphase plate, where all of the chromosomes line up in the middle. And that's it. So that's an overview of mitosis and all the different stages and steps and most of the relevant information about the process. We really didn't go into very much detail about the regulation, but we will in a video I will make on the regulation of that cycle. So get ready for that. And actually, I'm debating whether or not to do a video on it or to have that be part of the extension process, which I might do and link it with cancer. So regardless, whether video or extension process, we'll talk about how this process is regulated, and then we'll move on to talk about how this can occur sexually to reproduce brand new organisms, and talk about how it can contribute to their genetic diversity and thus their variation and eventually evolution. Hope you've enjoyed, guys. Have a wonderful day. Stay safe and healthy.